Hello, welcome everyone. We're so glad you could join us this evening. I'm Emily Bell with the Bow Foundation, and this is my husband, Stephen Bell. A few things before we get started this evening. We're so sorry we're obviously not in St. Louis this past week at the GNAO1 conference, but we hope everyone is staying well during these unusual times at home. This is our second webinar. Last week, we were joined by the McNamara Habib family, and we had a wonderful discussion about inclusion. If you missed that, we plan to share a recording in our next monthly update, so keep an eye out for that. Also, I want to give a special thank you to Nicole Crump, one of our amazing GNAO1 parents and volunteers who helped plan a lot of the conference in St. Louis that obviously did not come to fruition. And a special thank you to Dr. Viehoever, Dr. Axeen, Shirley, and all of the clinical research team for their efforts planning the conference and clinic, and now replanning that to be remote. So they're working on that, and Dr. Viehoever will update us a little bit more. A few housekeeping notes today. The webinar is being recorded thanks to Dan Habib on here, and will be available later for those who couldn't join us right now. I will introduce our presenters, but then we'll give a more in-depth bio right before each of them give their presentations. Today we have Dr. Amy Viehoever, Dr. Erika Axeen, Dr. Richard Neubig, Dr. Brian Muntean, and Dr. Scott Baraban joining us. Each of them will give a presentation about 7 to 10 minutes, and then at the end of all of the presentations, there will be about 10 minutes left for questions. You can utilize, there is a chat function or a Q&A function at the bottom of your screens that you can use to ask questions. And for the panelists, when you're getting to the end of your 10-minute window, if you're getting over, I'll turn my video back on so you can see me, and that could be a cue to sort of wrap up if possible. So we'll get started with Dr. Amy Viehoever. Stephen, you want to introduce her?
Sure, yeah. Thanks again, everybody, for joining us. What I think we'll do is each panelist will have 7-10 minutes. As we get to the conclusion of that window, we'll turn our video screen back on so that the panelist knows that they're approaching the end of their window. Dr. Viehoever is going to kick us off. Many of you know her. She completed her MD, PhD at Vanderbilt and her child neurology and movement disorder training at St. Louis Children's Hospital and Washington University in St. Louis. She first took care of a patient with GNAO1 in 2013 during her movement disorders fellowship, and she's since gone on to be one of the leading authors in the first study looking at movement disorders triggered by GNAO1. She's currently leading the natural history study of GNAO1 at WashU, and she's working to try and gather data about the natural history of the GNAO1 disorder to build the foundation that can lead to future clinical trials. So we'll kick it over to Dr. Viehoever and let her kick us off.
Hi guys. Okay, and thank you. So I'm going to share my screen and then hopefully put my presentation on. Can you see my presentation?
Yes, that's working. Great.
Okay. So yes, so I'm not going to go into very much detail about the disease because most everybody here is familiar. But some of the hallmarks that I think are important to mention are that one of the things that we found from our research is that almost all children with this disorder initially have some sort of hypotonia or low muscle tone when they're infants and young children. This leads to some sort of developmental delay, although in many of the kids, especially those kids without epilepsy, we think that intellect is preserved. And then most kids go on to develop either an early epileptic encephalopathy or a movement disorder that can be very variable, but has a hallmark of producing these hyperkinetic movements or storms of what I call chorea, and some kids have both or all of the above. There's a very kind of, there's a genotype-phenotype correlation that is not terribly specific, but there does seem to be certain mutations in the gene that cause the epilepsy. and some mutations that tend to cause only a movement disorder. I'm not going to talk a whole lot about that, but I will say that when I'm teaching residents and other physicians about this disorder, I will tell them that, you know, in many cases, you know, the movement disorder can look like your run-of-the-mill cerebral palsy when you first look at it. But one of the hallmarks are these storms of movements that are not typical for other causes of cerebral palsy. And these chorea storms or movement disorder storms can be characterized by these very violent movements that lead to muscle breakdown and what we call autonomic instability. And when I am talking about autonomic instability, I mean that there are fluctuations in blood pressure, in pulse, in temperature. Kids can look like they're running a fever when they're not sick with any kind of infection. And this can be life-threatening in many cases. Okay, so when we proposed to run the natural history study to the Bow Foundation, we described 4 aims that we wanted to pursue. The first was to kind of define the clinical phenotype, or get a definition of and a description of what kids and young adults and look like with this disorder. We also wanted to be able to gather data to prove that we could produce information such that other institutions such as the NIH would be willing to fund us in the future to do clinical trials. In that same vein, we wanted to start the work on developing a rating scale such that when there might be clinical trials ready to go to test, that we would have something to measure, that we could show whether or not the clinical trial is working or not. And then we wanted to be able to generate an initial set of recommendations for other doctors and nurses around the world on what are the best recommendations we have for treatment for this disorder. And actually, you know, when I initially presented this to the Bow Foundation, I thought that aim 4 would be the easiest thing to come up with, and it's actually proven to be one of the more challenging components. So for those families of you that were at the inpatient conference and research gathering last year, you'll recognize that we took a lot of data, right? So we asked a lot of families, and we did measurements and screenings and questionnaires, and trying to do a very big capture of information to try and help figure out what is going to be valuable moving forward. So I'm not going to go through all of these, but I want to thank all the families for going through all of this. So I'm going to present just a few of the results that we're planning on publishing in a manuscript upcoming. So the first thing that I think was surprising to me, at least, and to other researchers, is how varied the spectrum was of motor disability in GNAO1. So all we knew before this conference was what had been published in the literature, which were these very severe cases where kids were very severely disabled. Almost all of them would qualify into the category of GMFCS level 5, which is completely wheelchair bound and not able to do any care at all. And what we found is that actually there's a much bigger spectrum of disability, from everything from someone walking along the street that you would never know had a disability to our kids that we knew that were wheelchair bound and needed complete care. And so one of the things that we looked at that's very important to me is quality of life scales. So we took a quality of life scale called the CP Child, which has been validated in cerebral palsy, and looked at that compared to kids who have typical cerebral palsy, right? So the overall scores compared very similarly to other studies of kids with cerebral palsy. And I'm not going to go through these numbers in detail, but one of the things that I wanted to point out that was very interesting to me, and I think most of the parents who I interact and families that I interacted with would kind of agree with this, is that if you look at it with finer detail, what you'll notice is if you break it down by section, and here this graph is showing the scores with 100 being completely normal, as the level of disability goes up. And so you expect these scores to go down, the quality of life measures to go down as the level of disability goes up, okay? And so remembering that a GMFCS level 5 is completely wheelchair bound and unable to help with any level of care, and a level 1 has minor deficits. If you look at this graph, I'm going to use my arrow, I don't know if you guys can see it, but as you would expect, as the level of disability goes up, you see that levels of personal care, daily activities, positioning, transferring, those all suffer as the level of disability goes up. But what is different in kids with GNAO1, at least in this cohort, compared to other children with similar levels of disability with cerebral palsy, is that comfort and emotions, seen here in the green, communication, quality of life. If you look at these levels of the level 3s and level 4s, suffer much less than the overall mobility measures. And that is really interesting to me and kind of correlates with our understanding and our experience with the kids with that, which is that these kids seem to have much less emotional dysregulation as compared with other kids with movement disorders, and seem to be, in generally, happy kids with relatively low levels of anxiety, which is very interesting from my perspective, especially in understanding the pathophysiology of the disease. This is also another very busy slide that I'm not going to ask you to completely look at, but I did want to kind of point out a few things that are going to be important moving forward. The first all is that these are all different measures that we did, and if you look at these bars are for the patient that had a very mild level of disability, and these bars would be relative to a patient or a participant that had very high levels of disability from a movement standpoint, okay? And you'll notice that the GMFM, which is a measure of movement, okay, goes down as would be expected. These other measures of, say, spasticity and dystonia do not decline as you would expect, and remain about the same, indicating to us that maybe these are not the best measures for our population as far as figuring out what is an important measure moving forward, okay? So for the future, so we are going to be collecting data remotely. We are going to move forward, and we're going to have sessions where the physical therapist directs children or young adults to perform certain activities. We're going to gather the data. I'm going to be a part of that. I'm also going to conduct video visits with especially new families to collect medical history and things like that. We are encouraging as many families as can participate to participate, even if you didn't participate last year. I really feel like we can learn from every child or young adult or older adult. We're going to be sending out some new forms to try out, both in hopefully taking advantage of this opportunity where everybody's at home to log events and log things like seizures and movements and sleep and stuff like that, and testing out different scales that we want to try and use in the future for clinical trials. And then hopefully this will all lead to publications that we can use to gather funding to fund a much bigger study. So that's all I have for right now.
All right, thanks, Amy.
Thanks very much. That's a neat snapshot into some of the research that's already taken place and some of the initial findings you've had. Looking forward to continuing to have that grow. Our next presenter is Dr. Erika Axeen. She's a pediatric neurologist who specializes in evaluating and managing seizures and epilepsy in children. She has a particular interest in clinical diagnoses and the management of genetic epilepsies. Dr. Axeen was born and raised in Ames, Iowa, and she studied biological sciences with a minor in art history during undergrad at the University of Iowa. She completed medical school at the University of Iowa before moving to Charlottesville for her residency in pediatric neurology. She then went on to do a fellowship at Boston Children's Hospital in epilepsy and epilepsy genetics training with Dr. Ann Fedori. And once completed, she returned to UVA where she is now, and she's partnering with Dr. Viehoever on some of the natural history studies that we just talked about. So Dr. Axeen, we'll turn it over to you.
Great, thank you. Can everybody hear me?
Yes, we can.
Yes, great.
So Dr. Axeen, will you get that pulled up and keep playing with it? We've got a couple questions that came in for Dr. Viehoever, so we may try to talk through those while Dr. Axeen works to share her screen, if that's all right.
Yeah, that's fine.
Some of the questions that came in, three in particular. One, are there any similar illnesses which don't have specific genotype, phenotype correlation, similar to GNAO1? Is there anything that pops up in your mind like that?
It's a hard question to answer. I'm
Yeah,
sorry.
You stumped the expert.
So yes and no. And I might ask to come back to that question another time so that I could come up with specific examples to explain to people what I mean. And Dr. Axeen could probably help with that as well.
I think you might be able to address some of these other questions. This other question is to ask about, have there been any measurements on sleep in GNAO1 patients, which I think that is a part of the natural history study, if you want to talk about that.
And so one of the things that we're going to be doing in sending out to the families is we're going to ask you all, and hopefully you guys will agree, is for a 7-day log. And one of the things that we're going to ask you to log is sleep. And I've been in talks with one of my colleagues who has been a part of other research studies where we've looked at genetic diseases, and we've looked at sleep and doing sleep studies specifically, and what that would look like. So that would probably be part, not this year, probably not even next year, but maybe in future years, of probably doing sleep studies on a subset of patients. And for those kids that have had sleep studies in the past, getting those records to try and evaluate that and understand whether or not there's anything unique about sleep in GNAO1, whether or not it's just related to developmental delay and other kids who have, say, cerebral palsy or epilepsy, and whether or not intervening on that would be helpful.
All right, Dr. Axeen, you're going to try again?
Yep, I think I got it.
Perfect.
Are you able to
Thank
see?
you.
Sorry, that was so laborious. So hello, everybody. Most of you have probably met my colleague, Dr. Goodkin. He sends his greetings. It's his birthday today, so he's having a good time doing something else. But I am pleased to be here to share some of the results of the GNAO1 registry. I'm hoping that most of you here have already heard of this, and for those that have loved ones affected by GNAO1, you're already involved in this. But I'm just going to share some of our initial data here. So briefly, I'll talk about the registry design, just some basic demographics, medical history, touching on some basic features of the seizures and movement disorders, and then talking about next steps. So the registry aims, many of you have probably already seen, but really just to describe what is GNAO1. So a lot of the things that I'll share with you guys today, those that know people with GNAO1 related disorders, you'll say, well, yeah, of course that's a feature, but making sure that that's described in the literature is very important, so that we can target it for research. Also, having information available so that children that are newly diagnosed with GNAO1, the families are able to get good information about what to expect for their child. And then thirdly, to develop a registry of patients affected by GNAO1 to help pair them with researchers should there be research studies. So hopefully most of you have seen this page. This is the registry landing page at Invitae. And this study was sponsored by the Bow Foundation. So just briefly, the survey was put together by myself as well as with input from those in the Scientific Advisory Board. And the data were de-identified and IRB was used. The data that I am going to present was collected up until December 2019. Data collection is still ongoing, so but we had to pull the data at some point. So that's what I will present today. And this is a parent-generated survey, right? So this is different than a lot of what's in the literature already, which is with medical records. And what we have so far is 63 completed. There were 86 started, so if there's anybody on this call that thinks they might not have finished their survey, I'd encourage you to log back on. But we do have 63 data on 63 patients, which is wonderful. So this is truly a international effort. So we have patients involved from 5 continents. You can see the majority are North America and Europe, and even though there is a Google Translate option, I suspect language barrier is a big part of that. This map is not entirely accurate. Not everybody from the US is from LA or New York, but it's just kind of showing the distribution in the world. Over here, these numbers, these are in order of number of participants, and most of the ones here have low numbers, whereas the US are higher, and the Germany, et cetera, are medium amounts. So just briefly, we did collect genetic data, but we're limited a bit by the way that we collected it, and that we weren't able to just have fill in your own information. So it was a drop-down option, so variants that are already reported in the literature were included, with the exception, I believe, of a few of the microdeletions that may have been missing. But what we see is that the hotspots are well represented. Only 15 out of the 63 were not able to actually find theirs. So the majority of people were able to report their variant. So that will be helpful going forward. And for those that were disappointed that their variant's not listed, we're hopeful that as Invitae continues to update things, we'll be able to get that data as well. Here you can see that the sex of the participants was pretty even. So that's nice to know that in a large cohort, 63 patients, that it's pretty even between girls and boys. Down below here, you see the ages, and kind of as you would expect, given that this is a disorder that's only recently described, the majority of those that are entered in the registry are under age 10. And that's not to say that there aren't many people that are in their 20s or 30s out there. It's just that they might have skipped the genetic testing and they may not have the diagnosis. So just touching on some of the highlights that we have. I thought this was very interesting, and Amy already touched on this, but what is this, what made you first concerned about your child, and what age was it? So this talks about what age, and interestingly, every single respondent out of 63 indicated that within the first year, they were concerned about something. We'll talk about what that is in a moment, but here you see the raw numbers here that 22, 23 within the first 6 months. So percentage-wise, 73% of those, there was something different within 6 months, and then it goes up slightly to that 100% by age 1 year. So what was the first symptom of concern? This question, you were able to select more than one answer, so that's why the numbers don't add up to 100. But 70% reported hypotonia or low muscle tone. A pretty similar number, 67, reported failure to meet developmental milestones. Those things are often intertwined. If you have low tone, it's harder to do things like hold your head up, etc. And I think that that probably is also closely related to the difficulty feeding here in 24%, which I'll touch on again. But you also see the prominent features that we've been talking about with seizures and 25% as the first symptom of concern, right? So that might have been in the neonatal period or as infantile spasms. And then you see the abnormal movements, and that number is lower, even though, as I'll show you, the incidence of movement disorders is higher in this population. And I think that relates to when these symptoms start. And I'll show you some data about that. One comment is that although the majority of these patients did have hypotonia, there was a question that queried whether hypertonia, high muscle tone, was present, and 19% of respondents did report hypertonia. So, and that's pretty common, and I suspect that's related to the dystonia findings, but mixed muscle tone is something that I'm sure Dr. Viehoever is interested in learning more about in the study. So feeding difficulties in 87%. Now, just asking, have you had feeding difficulties is pretty non-specific. Many newborns struggle, but 49% reported feeding difficulties beyond the newborn period. And 38% have required a G-tube, which really speaks to the severity of the feeding difficulties. My pet interest, seizures and epilepsy. In this cohort, we found that 49% reported a history of seizures. So that could be seizures in the past or present, the way the question was worded. But 48% had never had a seizure. As expected, or not surprisingly, 2% did say they weren't sure. And I think that's related to sometimes the ambiguity of trying to distinguish between seizures and movement disorders. And then are they still having seizures? Actually, of those that had seizures, so 15 participants said, yes, they're still having seizures. That ends up being about 24% of the total surveyed say they have continued seizures, so likely active epilepsy. But I think encouragingly, and what's speaking to what Dr. Viehoever was saying about the spectrum may be broader than we expect in epilepsy as well, is that 16 participants said they don't have seizures anymore. They've had some seizures. And I think this is what it needs to be described more fully as both the severe end of the epilepsy with the developmental and epileptic encephalopathies, but those patients that have some seizures and they outgrow it, because that's certainly something that families would want to know as their child is developed with epilepsy. Regarding the movement disorders, the higher incidence of movement disorders in this cohort, 76% reported that they did have a movement disorder, but a much higher incidence of unsure, the 13% or 8 participants. And I think that maybe, again, a little bit of wording, what is a movement disorder? It was described a bit, but I think still it's kind of a complex thing. And the question of is it seizure, is it epilepsy, is something that I struggle with on a daily basis. So I'm sure other patients do as well. Additionally, the types of abnormal movement. So, this is a parent survey. So understanding the intricacies of the differences between chorea and athetosis is, I'm sure, something that Dr. Viehoever is great at. I'm an epileptologist. I'm not so great at that. So expecting parents to know the difference, even though I know that the GNAO1 parent groups are very well educated on their children, it can be very challenging. So I take this data with a little bit of a grain of salt, but I do think it's interesting. 47% said that their child has dystonia, 44 chorea, and you can see it goes down to tremor being just not as prominent of a feature in this population. Another way of looking at this is, this is a little bit of a complicated slide, but over here on the y-axis we see the number of participants. Down here is the age. So looking at what age do they start to have their abnormal movements. I mentioned tremor is not so much of a big deal in this group. So tremor is here in the green, and you see that's the clear outlier that really not reported, a handful over age 10, but not so much a concern. You see the rest of these all kind of grouped together. And I think what is most interesting here is that they kind of follow the same trend lines. You see, you know, there's less bulismus, which is the wild flinging of a body part, as compared to dystonia, right? So this is 15 participants versus 30. But if you think about when do 50% of these patients develop those first symptoms, all of that is at a young age. And that's really, you see this rapid rate, rate of increase here is pretty steep with all of them, and kind of by age 3 and even by young school age, over half of them and the majority of them have already presented, although there's a few that develop symptoms later. So this is something I'll dive into a little bit more, but I think is interesting. So overall, comparing some of our rates to published data, the Kelly paper in 2018, I had the honor of being part of that with Dr. Padouri, where they actually had medical records, and I'm sure some people on the call have children that are involved in this, or that are in this paper, but they went through medical records and reported all this. The registry, of course, is parent initiated. So the rate of seizures and epilepsy is lower, only 49% in the registry as compared to Kelly data. I think that may be related to some bias, in that people that are seeing Dr. Padouri, Dr. Sheffer, and others are who are epileptologists. That's why that's going to be higher in that. But I think also speaks to the need to describe the milder end of the epilepsy spectrum as well. The movement disorders, shockingly, is exact same number, 76%, which suggests to me that that's probably about accurate. And then thinking about which patients have both, and they reported 42% in this cohort. Just looking at it very simplistically, of people that said yes, it was 30%. But remember that those groups that said unsure for either the epilepsy or seizures or the movement disorder, if you include those, it gets up to 44%, which is, I'm sure, an overestimate, but is a little bit closer to this number. So the truth probably lies somewhere in between there. So in summary, I think this does show some things that, again, I'm guessing that those who are listening see these features and say, yeah, that's us. But what we can see here is that the symptoms presented before age 1, low tone hypotonia and feeding difficulties were very prominent. A history of seizures was seen in half, and 24% with continued seizures, and that movement disorders are more common than epilepsy in this population. So this is all unpublished data, so the next steps will be getting it published. This registry is open, so if anybody hasn't signed up, please do. And then my specific goal is to describe the epilepsy features a bit more. So we're hoping to do a focused survey on that patient population as well. So if anybody listening has a child or participant with epilepsy, keep your eye out for that. And this is a beautiful picture of the mountains that are near where I live. It's the reason why I left Iowa and live in Virginia. So, all right, thank you.
Thank you so much.
Mm-hmm.
It was great that the first chance I had to hear some of the readout from the survey we'd done. That was neat.
Mm-hmm.
Thanks for spending time with us, Dr. Axeen. That was insightful.
Mm-hmm.
Our next presenter is Dr. Rick Neubig. He received his Bachelor of Science in Chemistry from the University of Michigan and his MD from Harvard in pharmacology from Harvard University. He trained in internal medicine at the University of Michigan Hospitals, then joined the faculty in the departments of pharmacology and internal medicine. He went on to become the chair of pharmacology and toxicology at Michigan State University in 2013. Dr. Neubig has studied GPCR signaling for most of his career and currently explores G proteins in genetic epilepsies. So Dr. Neubig, thanks for joining us. We'll turn it over to you.
Hey, hello everybody, and let me get my screen up there. Can you see the screen?
Yep.
Yep, great, thank you.
Okay, very good. So I'm going to give you an update on our research on the GNAO1 mouse models. And I wish I could give a very clear picture, but the picture is unbelievably muddy and confusing. I will be talking in part to the researchers so that they can really pick up and try to move forward in answering some of the questions that are really a challenge to us here, trying to correlate the animal models with the clinical spectrum. Okay, get over on this screen. So as you let's see. So as you know, G protein or GNAO1 encodes a protein that carries signals from receptors on the outside of a cell to effectors on the inside of the cell. And one of those effectors, one of the signals that occurs is something called cyclic AMP, which is a second messenger that carries signals inside the cell. And when you have a mutation in a signaling protein, there are two general flavors of those. One is loss of function, where the protein is damaged in a way that causes it to signal less. There are also options, these are very common in cancer, where you can have gain of function mutations, where the mutation changes the signaling protein in a way that makes a signal stronger. And so that's one key factor that we've been looking at as we first started our studies of GNAO1. And that was done by work by Jade Fang, a PhD student in my lab. And when she looked at a lot of the mutations and asked what their function was in a cell system that we were studying, many of them had gain of function mutations. Some of them had loss of function mutations. And what we found was that the movement disorder, patients with movement disorder tended to have mutations that led to gain of function. And patients who had the epilepsy generally had mutations that led to loss of function in this cyclic AMP assay that we were studying in the lab. There was one mutation in particular, which is really fairly common, the G203R mutation, where the patients very frequently see both the movement disorder and epilepsy, although I believe the movement disorder is probably the most bothersome to patients, and the epilepsy is in many cases relatively well controlled. So that dichotomy that movement disorder comes with gain of function and epilepsy comes with loss of function seems to hold in the clinical pattern. And one of the things we were trying to do in my lab was to generate mouse models that would carry some of these human mutations to be able to do studies for drug testing and things like that. And the bottom line is we've had a very difficult time generating these models. So we started out making a model of the G203R, with which biochemically has the gain of function and clinically has both the epilepsy and the movement disorder. So this particular mouse, we did see the mice were viable. We had rare adult lethality, but they were generally pretty viable. They bred well, and they had a seizure and movement disorder phenotype. We also tried making some of the more severe epilepsy mutations, the in-frame deletion delta 191 to 197, and with the support of the Bow Foundation, we also made a G40R mutation. In both of these cases, the mutant mice were not viable. So, mice are not people, so we can't make direct correlations, but in these cases, the problems were so severe that we couldn't even generate a viable mouse strain. The other mutation we made with the support of the Bow Foundation was the R209H mutation. And biochemically, this had relatively normal function. Clinically, it has largely a movement disorder. And our mice were very viable. This mouse line is working very well. It breeds well. What we see is hyperactive movements, which is consistent with the general pattern of hyperkinetic movement disorders, in the patients, but I think the specific pattern we saw of hyperactivity does not necessarily correlate perfectly with the clinical pattern. And we did see suppression by risperidone. I'm going to show you data on this mutant, and then get into what's making things very complicated. So with the R209H mutants, what we saw was that the heterozygous mutants that carry only one copy of the mutant gene, which is what most of the kids have, were quite hyperactive. You take a mouse, you put it on an open field, and then it gets tracked where it goes, and the mice were very hyperactive. They traveled much longer distances. And what we did see was that a drug that has been used to treat the kids, risperidone, which blocks dopamine receptors, can suppress those hyperactive movements. And that was, unfortunately though, it also affected the normal mice as well as the mutants. So it wasn't selective for the mutants, which is what we would hope to see. So that these mice are available. We have them in our lab. We are planning to submit them to Jackson Labs to make them available to other researchers if they want to study them. And the paper was published in just a couple months ago in a journal. So the other mouse mutant, the G203R mutant, we published this a while ago. They have a movement phenotype. We put them on a moving belt, and their stride length is lower. So they take shorter steps, but they have to take more steps to keep up with the belt. They also would fall off of a rotating rod, indicating their balance or their ability to respond quickly to movements were not good. And so when we look at these two mutants, what we see is the R209H has hyperactivity and a little bit of a decreased stride length. The G203R had reduced balance on the rotor rod, decreased stride length. They also had increased seizure susceptibility. So this was consistent with the idea that the patients have both movement disorder and seizures. So what's confusing though is when we try to connect everything with the gain of function, loss of function, and match everything up, we had these two clinical mutants with loss of function that were very severe, and the mice would die early. There was a mouse also made in the literature where it was a design knockout. It completely knocked out the gene, so that would be a complete loss of function as a heterozygote. But those were viable and basically had very little in the way of seizure or movement phenotype. So even though these three were all loss of function biochemically, they had very different phenotypes, which may suggest that these mutations are not pure loss of function, but have some other function that they have gained. So there's new functions that can be gained in some cases. So that's a confusion that's been in my mind and in the literature for quite some time. We also made another mutation. The first G203R mutation we made had a mouse sequence in a particular region. And to be able to allow gene knockdown studies, we wanted to make a humanized G203R. And with help from a patient group, we were able to make those mice, target them with CRISPR-Cas9. But unfortunately, we tried 3 times with a CRISPR, and we never got a viable mouse strain. We did get some mutations, but they tended to be low penetrance. It was a low frequency of the mutation, and we never were able to generate a breeding colony. So we had a big confusion here. We had confusion between these not matching this, and we had a confusion between this not matching this. So we actually went back and looked at our original mouse that we had published on. We had made this mutation, the G203R mutation, in the sequence. But when we went back and looked at it again, we discovered something we had missed the first time, that there was a second mutation in our mouse G203R strain. And what that does is it actually changes a splice site, and this is very technical, changes a splice site so you don't actually ever produce the mutant protein. So even though we thought this was a gain-of-function allele in our original publication, when we went back and found this unfortunate second mutation, it really now looks like this is not a gain-of-function, but is a loss-of-function. And so when we go back here, the milder phenotype of this mouse matches more with the knockout. So again, I understand this is likely very confusing. So what we're seeing for GNAO1, the genetics are very complex. One type of a knockout or one type of loss of function is lethal. Another type of loss of function is not lethal and has very little phenotype. And so it's been very difficult because of this, both with these mutations and now with this mutation, we've really been unable to generate viable mouse mutants. And so that is a very serious problem with trying to go forward with mouse studies. And it also clearly says that the loss of function, gain of function model, which does, in my view, still correlate pretty well with clinical patterns, really can't explain the mouse models. So this is a fairly confusing situation and is making it very challenging to go forward. I'm hoping to hear from Dr. Baraban about some of the other model organism studies, which I'm hoping may shed some light on these very complicated results that we have to date. And so I need to thank especially Jade Feng, who did the work in my lab as a PhD student. Cassie Larive was a master's student who worked on the R209H study. Jade is now a fellow in an epilepsy genetics lab at Children's Hospital of Philadelphia. And then we have the funding, including the funding from the Bow Foundation, which was very valuable to be able to learn what we have been able to learn from the mouse models, even though they've been challenging to create.
Dr. Neubig, we actually heard from a family today that they met Jade recently, that she was able to sit in on their clinical visit. So we're excited to know that she was still connecting with GNAO1 families in Philadelphia. So we thought that was really great.
That's great. I'm glad to hear that also.
Yeah, well, thank you.
So our next presenter will be Dr. Brian Muntean. He obtained his PhD at the University of Toledo, focused on studying the core concepts of GPCR signaling in genetic disease models using cell culture, biochemistry, and in vivo imaging of biosensors. He then joined Dr. Kirill Martemayanov's laboratory at the Scripps Research Institute for postdoctoral training. There he studies the genetic aspects of neuronal signaling in native tissues. The main themes of his work include understanding the complex GPR signaling systems, how those contribute to regulatory networks towards pathology, and discovering small molecules that adjust signaling dynamics. Dr. Muntean received a grant from the Bow Foundation earlier this year as a postdoctoral fellow, and we appreciate him joining us today to give us all a little bit of an update and just tell us what he's working on.
Thank you for the introduction. Can everybody see my screen?
We can, yes.
Okay, I can't see anybody else, but I can see my screen. So unless you tell me otherwise, I'll assume that you can see and hear me fine, because I can't see anyone right now. Okay.
Yeah. Great,
So
go ahead.
okay, great. First and foremost, I'd like to thank the Foundation for awarding me the postdoctoral fellowship, which really supports our work toward understanding the GNAO1 pathophysiology. And this is also going to help us raise awareness of this issue throughout the scientific community. Second, I'd like to thank the Foundation for organizing the webinar. I'm really honored to be here tonight and to also connect with the community. So what I want to do over the next few minutes is just give you a big picture conceptual overview of what I've been working on, what I'm planning to do, and what's the rationale for really motivating these studies. So due to the time constraints, I'm going to simplify some of the details and therefore reserve time at the end to expand on any areas where questions may arise. So as we know, the GNAO1 disorder is associated largely with the manifestation of seizures and abnormal movements. So these different behaviors are controlled through different parts of the brain. The research that I do is specialized on the abnormal movement aspect of the GNAO1 disorder. So in order to coordinate movement, the brain has to utilize several different brain centers. And amongst these brain centers, there's a large region of the brain called the striatum that's really important for initializing actions, selecting which actions we want to engage in, learning new tasks, as well as performing habitual movements. And this part of the brain is also abundantly expresses the GNAO1 protein G-alpha-O. So in this striatum is where I'm going to be doing all of my studies. And before we get any deeper, I want to say, however, that even though I'll be studying in the striatum, I think that any insight that we gain here is going to be highly valuable toward neurons in other parts of the brain, including the hippocampus or the cerebellum or the cortex. Okay, so I put some pictures on here, a little cartoonish of what the striatum looks like. And these blue lines represent different parts of the brain that send messages to the striatum. And what the striatum does is it receives all of these messages, integrates this information, and then uses that to send instructions to the rest of the body to tell us when to move, how fast to move, when to stop moving, how to learn things, this type of stuff. And these messages are transmitted in the form of chemical signals. So you may be familiar with some of these common signals that we've heard of before, such as serotonin or dopamine, norepinephrine, or glutamate. And I like to think of these signals analogous to sort of a Morse code. So if we zoom in on the striatum, I'd like to try to highlight that a little bit. So within the striatum, we have all these chemical messages that arrive at the cells. And as Dr. Neubig just kind of showed, we have different types of neurons in the striatum. And I've used colors to kind of simplify things instead of getting into too much jargon. So what will happen is these different chemical messages arrive at these striatal neurons, and then unique receptors on these neurons are paired to identify these different messages. And so what happens is once this receptor is paired to a unique message, and then it's up to the responsibility of the G protein machinery, such as the G-alpha-O, to actually transduce this signal and alter the striatal activity. And so it's actually these G proteins that decode this Morse code of signals that the striatum receives. And then the balance of activity between these different types of striatal neurons is what's used to calculate how we execute our locomotion programs and coordinate our movements. So in this context, we can see that these G proteins are actually this key mechanistic bottleneck that decodes these signals and drives our movement. When it's illustrated this way, I think it may make it a little easier to understand that we lose the ability to fine-tune the signal decoding process in the event that we have compromised G protein activity, which is unfortunately the case in the GNAO1 disorder. And so, building off of this, my hypothesis is that these GNAO1 alterations imbalance signal decoding processes in these striatal neurons, and that's what's impeding our locomotion. And so my proposal, there's two parts or two phases to the plan, and I do all of my work using a mouse model systems. And so in the first step, what we would like to do is to simply understand what's so special about the GNAO1 protein G-alpha-O and its role in decoding these signals and driving the activity in the striatal neurons. So to do this, we actually use these primary neurons from mouse tissue using live neurons and live brain tissue in these mice. And so in doing this, we're hopeful that any results that we gain will be more translatable than some other approaches. And also by sticking with the mouse system, this enables us the opportunity to alter the genetic background of the GNAO1 gene. So after we can uncover precisely how this G-alpha-O is operating in these neurons, we can then begin to plug in these different mutations in the neurons and see how these mutations impart abnormalities in the signaling. So that's going to be the second step of the process. And so how this is actually done is going to be largely using a technique and technology that I've spent the past 4 years developing in Dr. Kirill Martinyanov's lab in Scripps, Florida. So what we've been able to do is create a genetically modified mouse that contains a biosensor that reports the activity of these G proteins in real time in the neurons. And so we do that by taking these live neurons and these live brain tissues, and we actually quite literally look at the activity of these neurons using different microscopy approaches. So we can visualize and illuminate what's going on in these neurons. And we're doing that right now in tissue with manipulations of the GNAO1 genome. And so what we think this is going to enable us to do is identify essentially which receptors are misbehaving because of these different mutations. And so moving forward, if I come back to this slide again, what we're hopeful to do, and this slide is a little bit intentionally complex, so we actually want to harness the complexity of this system, of these decoding mechanisms, to actually rebalance the activity in the striatal neurons in the GNAO1 patients. And so the long-term idea is that if we can uncover which receptors are signaling differently through these mutations, and then we can counterbalance the activity. So in an abstract example, if, for example, the red receptor is signaling too strongly, perhaps we could counterbalance that by activating the orange or the yellow receptor. Or on the other hand, if we don't have much signal from a green receptor, perhaps we could augment that from this blue or purple receptor. And so a real world example of that in a different pathology, Parkinson's disease, where it's a different motor deficit. However, the problem is largely because of perturbations in the dopamine system. And so we think something like that may be going on, whereas in Parkinson's, you can augment the dopamine and have a short-term efficacy in the patients. We think that there may be strategies using pharmacological agents to selectively alter different receptors in these different neurons and then rebalance the activity in these neurons and hopefully have a chance to alter the locomotion program. And so that's kind of a big picture overview of what I'm doing. And I get to do this work every day with a very talented team. And that starts at the top with my mentor and the PI of the lab, Dr. Kirill Martinyanov, as well as several other really talented personnel that help me to perform these studies. And we've been fortunate to have funding from the Bow Foundation as well as the National Institutes of Health. So thank you for your time, and any questions I would love to hear.
Thanks very much. That was great. We're looking forward to hearing more as you continue your project. Shifting gears, we're going to turn next, and our last presenter is Dr. Scott Baraban. He's a professor of neurological surgery in the William K. Bowes Endowed Chair of Neuroscience at the University of California in San Francisco. There, he directs the Epilepsy Research Laboratory in the Department of Neurological Surgery, a research program continuously funded by the NIH for nearly 20 years. Dr. Baraban studies the cellular and molecular basis of epilepsy with a focus on catastrophic epilepsies of childhood, and his lab was the first group to use zebrafish zebrafish models of acute seizures and epilepsies starting in 2005. He's since gone on to publish several manuscripts describing how to evaluate seizure activity in larval zebrafish, the first zebrafish models for genetic epilepsy, and drug screening using zebrafish models based in epilepsy. His projects are identifying new treatments for these patients, and Dr. Baraban received a Bow Foundation grant earlier this year as well to create a GNAO1 specific zebrafish line. Looking forward to hearing from you today, Dr. Baraban. Thanks so much for joining us.
Thanks. Okay. Okay, thank you for having me, and it's been really interesting to catch up and learn more about the clinical side, because that really plays a big role in how we design our animal models, and similar to Dr. Neubig's lab. So I want to talk to you about zebrafish. I'm guessing not too many people realize that zebrafish are actually used for disease-based research in epilepsy and neurological disorders specifically. The reason we kind of turned to zebrafish some 15, 20 years ago was to develop a alternate system to do basic science research in the epilepsy field that would allow us to do eventually in vivo drug screening and in a high throughput manner compared to how we normally discover drugs. And the reason why I chose zebrafish has to do with a lot of features that it already had in the neuroscience community as a tool for a number of developmental labs. First and foremost, zebrafish are vertebrates. They have a very similar genetic identity with human. For all genes, it's roughly about 70%. For disease-causing genes, they have a sequence identity closer to 84%. So actually very comparable to what you think for mouse models. However, they're much easier to maintain. What you saw on that first page was just adult larvae that we keep in fish tanks in a lab. And we work on the adult zebrafish. And what we work on are the larvae, which you see in the top right, which are very tiny, about the size of your eyelash. And all they really require are fish tanks, controlled water setting, obviously, and fish food. And so it's very cheap to maintain thousands of zebrafish compared to rodents. They're genetically tractable, and we've taken advantage of this over the years to generate specific knockouts of genes for specific epilepsy genes. And most importantly, if you look at the larvae, you can get lots of them. Each week we can breed fish and get 100 or 200 larvae per plate that we can do experiments on that week. And whatever you add to the bathing media that the larvae are in, they'll directly absorb. So they're very efficient for doing drug discovery. So this, of course, is not something that came completely out of our lab. The adaptation to epilepsy did, but many labs around the world have used zebrafish for many years. As I said, initially they were used for developmental studies. Christian Nusslein-Volhard won a Nobel Prize for it some 20 or 30 years ago. More recently, people have turned to using them for drug discovery. And this was highlighted about 5 years ago in a Nature Reviews drug discovery paper by Randy Peterson and colleagues that focused on the idea that the targets are conserved, in this case, the genetic similarity between the gene and humans, that metabolism pathways are conserved, that the pharmacology is conserved, and that the physiology is conserved. And more recently, coming back to resurvey the field only 4 years later, Megan Culley had a very nice review that highlighted about 6 or 7 projects, including ours, where we've already found drugs in zebrafish for diseases that are in clinical trials. And that's kind of what I want to talk to you about briefly here. So the main issue we've had in the epilepsy field is that there's about 30 anti-epileptic drugs, or AEDs, that have been approved by the FDA. And these were first discovered back in 1850, and they continue to be discovered today. But what you can see on the little chart in the inset is that even though phenytoin was discovered in 1950, and another 20 some odd drugs have been discovered since then, the number of patients that remain basically untreatable by these drugs has maintained steady at 20 to 30%. And there hasn't been much change in this over the last 50 years. And one of the reasons I believe this is the case has to do with how these drugs are discovered and designed. First of all of these existing drugs came out of adult models, not genetic models of epilepsy, and not pediatric models of epilepsy. And they were all based on acute induced seizure episodes. So not spontaneous seizures, which is what epilepsy is in patients. These are low throughput approaches. So even if you want to screen for drugs in a rodent model, you might be able to do 10 or 20 or 50 a year. And we can do that basically in zebrafish in 1 day. They don't model spontaneous seizures because that's very difficult to monitor mice over time with video EEG monitoring and very, very low throughput. And so they're not really looking at epilepsy or anti-epilepsy drugs as much as they're looking for anti-seizure drugs, which is very different. Again, they don't target genetic forms of epilepsy, so none of these drug discoveries have focused on any of these rare diseases, and they're both costly and labor intensive. And so as an alternative, we proposed to use zebrafish about 6 or 7 years ago. One of the main reasons, of course, was going to be a lot cheaper to screen many drugs using zebrafish larvae. The time required, of course, would be cut dramatically down. In our case, I'm going to show you data that's taken us only about 4 or 5 years to gather. The drug quantities per assay is very small because we're putting these in 96-well plates, as you'll see. We only need a little bit of drug to see if it works. So we don't have to manufacture or obtain expensive drug candidates. It's high throughput. We can screen about 100 fish per day. And we've developed a very sensitive 2-stage assay, which I'll show you in a second, which includes both the behavior, so the spontaneous convulsive behaviors an animal has, but also more importantly, as a second screen, the electrical activity in the brain, because a true anti-epileptic drug should suppress the EEG activity back to normal. And as you'll see, this is a form of precision medicine that we could apply to any genetic form of epilepsy. So what you see here is how the assay works very, very briefly. The idea is that we screen drugs in a blinded fashion. We buy commercially available libraries, in many cases containing repurposed drugs, so that we can quickly go to the clinic if we find something interesting. We can screen 6 fish per drug, and you can see them, these little wells kind of swimming around slowly. And for each positive hit, which is basically in this first assay, a drug that makes this very convulsive behavior go back to a control level, which is very little swimming or no convulsions, we can quickly assess a change in their actual seizure behavior. We can repeat this. We'll track at least 2 or 3 times on the same drug if we find a hit. And then we can have a very quantitative analysis of this movement based on the software we use to track the zebrafish larvae's movement in these individual wells. So that's stage 1. But again, there could be a lot of false positives that come out of a strictly behavioral assay. You can quickly imagine that a sedative drug or a muscle relaxant is also going to cause the fish not to swim very much and reduce their activity back to a control level. But if the drug is truly anti-epileptic, it would also have to stop the seizure events. And so we developed techniques where we could actually record EEG from larvae that are live, embedded in agar. And what you see here is an example of one of those recordings. So this is a Dravet syndrome mutant. These are spontaneous electrical events that we associate with these seizure behaviors. And you can see after the application of stiripentol, which is a known AED that's given to Dravet syndrome patients, there's a dramatic suppression of these abnormal electrical events. So this gives us a very sensitive assay to first screen drugs on behavior very quickly, and then follow it up with EEG recordings. And this, again, would be very difficult to do if you had a mouse model. And so the proof of concept we started with was something called Dravet syndrome. I'm guessing many people in the community have heard of it. It's one of the most common genetic rare epilepsies. It's associated with a sodium channel mutation, so a voltage-gated sodium channel that controls how neurons fire. This loss of function mutation is seen in about 80% of patients in this SCN1A gene. It's a catastrophic childhood epilepsy, similar to GNAO1. It also has an early onset, usually during the first year of life. And as the children grow older, they have intellectual disabilities, social impairments, and a very high risk of SUDEP. And most importantly, like a lot of these epilepsies, these rare genetic epilepsies in children, they're resistant to these available pharmacology. So the AEDs that we have, like I said, which were developed for adult models or adult patients, don't work very well in these patients. And so there's a great need to find drugs quickly for this population. And so we designed and developed a loss of function mutant of a gene that's about 77% similar to the human gene. It's called SCN1LAB. You see it in the top left. So for context, this is one well of a 96 well plate, and this is one fish slowed down actually because its seizure behavior is very rapid. The video is slowed down so you can see the whole body convulsions that it has. A normal wild type fish will move very little and will never have these type of full body convulsions you see here. So these seizure events, as I said, can be tracked using a video camera. Again, a control fish might move a little bit in this well during a 15-minute recording, but a mutant fish is going to have a lot of these events over 15 minutes. And so that's our first assay. This is our behavioral assay, and we can quantify how many seizures this larvae is having in this well. We can then take that fish out, embed it in agar, and record a local field potential, which is what an EEG basically is. And you can see that, again, these have spontaneous electrographic seizure events that we could easily monitor. The interesting features of this Dravet syndrome fish is that it mimics many of the features of patients with Dravet syndrome. In addition to spontaneous seizures, these larvae die early, similar to the SUDEP risk. They have both developmental delays, they have sleep disturbances, they have metabolic delays, and they're sensitive to the same types of AEDs that are used as standard of care. So if we give drugs like stiripentol or fenfluramine even, or valproic acid, we can reduce some of the seizure activity. So they're very predictive potentially of what compounds would be useful. And so using this model, we've screened to date about 3,500 drugs. And again, this is a very relatively short time scale. This is just my own lab. It's not in collaboration with anybody. And it's basically with one technician screening once a week. So we screen about 100 drugs per month. And we've screened a variety of different commercially available libraries, as you can see here, natural products, repurposed drug libraries, more serotonin libraries, as you'll see, FDA-approved drugs, and more recently, some synthetic cannabinoids. From this, we've actually found 6 lead compounds during this time. All of them act on serotonin receptors, which we found more recently, basically using the fish to reverse engineer the mechanism. 3 of these compounds have already obtained FDA orphan drug designation because they're repurposed drugs that were already on the market for some other use. 2 of the compounds already have compassionate use clinical data, basically meaning some small patient populations have taken these drugs and shown seizure reductions. And one of them is now in phase 2 trials, which were just started last month. And phase 1, which was completely safe for this drug, was completed last year. This is a combination with Epigenics Therapeutics that is we work with to commercialize these discoveries we find in the lab so they can go quickly to patients. And importantly, we went right from fish to people. There were no mouse studies in between in these studies. And so very briefly to wrap up, we're doing something similar now with every known human epilepsy gene. We started with the 40 loss of function genes that are known, and we're making CRISPR mutants for each of those fish. We're raising them to an F3 generation or greater, and then we're going to use them, if they have pathological phenotypes, for drug screening and development. This is where the Bow Foundation funds will come in. This is just a list of all the genes and all the zebrafish lines we have already made. There's about 38 of them that are viable, and we're using them all initially to screen for epilepsy phenotypes. Basically using our electrophysiology assays, we screen every line to see if they have spontaneous seizures, because we want to look for drugs, again, in a drug discovery platform that would stop spontaneous seizures, i.e. epilepsy. We've screened 80 to 100 larvae per fish, per gene. We do all this blind, and then we post hoc find out what the genotype of that fish is. And so right now we have mutants, and this is our very brief plan for how we'll use the Bow Foundation funds. We've already generated loss of function F3 generation or greater lines for GNAO1A and 1B. There's gene duplication in the zebrafish genome, and now we're crossing them to make double mutants. We're evaluating all of them for EEG. We've already almost finished the A and B lines, and in a couple months we're going to start the compound lines, assuming the lab can open up again. We'll evaluate EEG first, because that's the more critical feature of whether the fish has epilepsy. And then if it has a spontaneous phenotype, we'll look at behavior, because that would be a readout that'd be amenable to drug screening, as you've seen, as I showed you. And of course, we'll test some AEDs that we know are out on the market that are used probably ineffectively in these patients to give us a background of what they already respond to. We can't promise, of course, that the fish will have epilepsy, just as Dr. Neubig knows with the mice, or whether they'll be viable enough to do these types of assays. But we do have a almost 2-decade experience making these models, developing these assays, and now doing drug-based screening. So we're hopeful that if we do get a line that's interesting, we'll be able to quickly move to the next phase, which would be to do drug screening on those fish. And I also want to thank the Bow Foundation, the NIH, of course, that supports the generation of the Easy Project fish, and the other foundations that have supported us over the years. Put this up if anybody wants to get in touch with us, my email, what the lab does in general, and to follow our kind of daily science academic Twitter. You can follow us on Twitter as well, where we post our research as well as others that are of interest to us. And so I think I'll stop there.
All right, awesome. Thank you so much, Dr. Baraban.
Sure.
One question before we open up. If anybody has any questions, Amy, you can unmute yourself, please. Yeah, you have a question.
So I'm just kind of curious, how are you defining loss of function? And oh, sorry, my kid's in the background.
Okay.
And then I also noticed that you had a couple of epilepsy genes on there that also kind of fall into this category of like mixed epilepsy movement disorder, so SBX, those types of genes. Right. And being that I'm a movement person, have you guys been able to distinguish like abnormal movements versus epilepsy in this kind of quantitative system that you have?
Right, those are 2 great questions. So first of all, they're all CRISPR generated loss of function. And so we target a region where we could eliminate the gene, and then we confirm that, of course, with sequencing. Knock-ins, which is a more common technology in rodents, is not as feasible at the moment for fish. So these are simply loss of function design CRISPR fish that we then breed out for a couple generations to get rid of off-target effects, obviously. The movement disorder is a great question. We have STXBP1 mutants. They actually have a pretty severe epilepsy, spontaneous epilepsy phenotype. What we're trying to do to separate out, I guess, movement disorders from epilepsy phenotypes, which is very difficult because remember, these are larvae. They have they can swim and have certain behaviors, but they're pretty limited compared to even a rodent model, and definitely compared to humans. So we're trying to set up to do higher speed imaging than I showed you, and that's why that video is slowed down. So basically, try to image them. So your video frame rate's around 33 frames per second. That image, the video I showed you, was 250 frames per second, and we're trying to get even higher to pick up more subtle movements. Infantile spasms are another one, because a lot of those lines are infantile spasms phenotypes as well. And we're working with Florian Engert at Harvard, who's developing his own, his kind of custom-made cameras and systems. It's a great question. I think it's one of the next steps we'll do, which is to look at the fine behavior of the fish, the fine movements in between these seizure events, because they're probably informative, but it requires some technical kind of leaps that we haven't yet figured out yet.
Dr. Baraban, one other question I had for you
Sure.
Before we move on to some of the other ones. And families, I apologize, I know we're running long. We're gonna ask a couple questions, and if anybody has any questions they want to put in the Q&A in the chat, we'll try to get to them as well, but we'll wrap up in a couple minutes. Dr. Baraban, you talked about sort of the process and the life cycle. For those of us who aren't as familiar with clinical trials and the phases of that, realistically, hypothetically, obviously, if you have success maybe in GNAO1 zebrafish and you find positive hits on drugs, when are we looking at that could be at the bedside, potentially helping some of our families?
Right. So it depends on what kind of drug we find. There's two possibilities. We find a repurposed drug, like you hear about with COVID right now with hydrochloroquine, a drug that's already FDA approved for some other purpose. And that was our case with 3 of the drugs that we used with Dravet syndrome. They were already on the market for something else years ago. That would go pretty quickly. Not a new chemical entity would take much longer because it would have to go through FDA steps that just, to be honest, take maybe 5 to 10 years. On the short end, if we found a repurposed drug and that we've already had this pathway with Epigenics on how to develop them, it would only take a couple years at this point because we've already kind of done this once and we're set up now to do it. It took a little bit, like clemizole was discovered, the first drug in 2013, and we're in clinical trials now in 2020. But part of that slowness was we didn't spin out a company to help us develop it until about 2018. So that's 7 years, which is relatively quick compared to a rodent study, from discovery to patient is really short. But I think that 7 years would be even shorter now that we've done it once. Again, assuming, of course, it's a repurposed drug that somebody has clinical FDA safety information on. So basically, we work with the FDA, and if it's a repurposed drug, then you kind of just go through these steps where they want to make sure the safety is okay and the data is okay. And then it's a matter of just getting the patients together for a clinical trial. We'd start with compassionate use because that gives us a very quick readout. That's basically patients volunteering who are without a placebo control group, just to see if they have a reduction in some of their phenotypes or their seizures specifically. So that could happen very quickly if we found something again, but it would only work if it's a repurposed drug.
Okay.
In terms of speed. I mean, the other drugs would be developed, they would just take longer.
Okay. Dr. Neubig, I have a question for you, if you can unmute yourself. We are talking a lot about animal models. Can you explain for some of our newer families the importance of animal models and why these animals are important to make this connection between Amy's work and helping our patients?
Yeah, so oftentimes what you can see in the animal models, if the genetics is perhaps less complex than we're looking at here, is that the animals will have patterns, behavioral patterns that are similar to what the kids or the patients will, and treating with drugs, and especially in the repurposing situation where you've got drugs that are already on the market for one disease, and if you find that those will help improve the phenotype in the animals, that is often an important thing to be able to move forward. And as Dr. Baraban mentioned, for repurposed drugs, the FDA really wants to see safety. And so you can, in some cases, bypass the animal systems. one thing you'd love to do would be to design in a computer or just do everything in a dish and say, okay, here we have a drug that changes the pattern of neuron function in a way that would treat patients. But the brain is much more complex than that, and having an integrated brain function, either in a zebrafish or in a mouse or a rat, is often needed to really better understand how the drugs interact with these complex brain patterns with lots of different receptors that are playing a role. So that often becomes important to really be able to make predictions. But as we learn, unfortunately, is mice are not just little people. There are clear differences, and the genetic pattern we're seeing in the mice is still rather confusing. Why the GNAO1 heterozygous knockout mice don't look like a loss of function mutant allele knocked into a mouse is a very confusing thing. It's been confusing me for about 5 years or 7 years almost, and I still don't have an answer to that. And I think the other thing you could do in a mouse model that may be more challenging in zebrafish is you can really begin to look at the interactions of different neuron types. So if you look at as Dr. Muntean mentioned, the striatum, the hippocampus, the different regions of the brain that underlie either movement patterns or seizure patterns, you can actually begin to understand the connectivities of the different neurons. And in that case, a rodent may be a better, it's not perfect model of humans, but it may be closer to humans in terms of what you can learn about those complex interaction mechanisms.
Yeah, Dr. Axeen, I have a question for you that came in on the chat. So one of the families was asking, is the condition always regressive? And I thought maybe you could speak to that briefly.
Good question. The answer is no, and that was one of the questions on our registry, and I can pull it up. I think it was something like 20 to 30% of families reported regression. So it was not everybody. And one thing I plan to do is look through that a little more detail and see if it's the epilepsy phenotypes that have more of that versus the movement disorder phenotypes. So the answer is no, not every patient will experience regression.
Okay. Dr. Viehoever, a question for you that came in via the chat. Have you seen a prevalence of circulation issues in GNAO1 kids? And this is something that you mentioned briefly in your presentation about dysautonomia. Probably saying that wrong. That's been talked about a lot on the family page. If you could speak to that a little bit, defining that for those who aren't familiar with it.
I think the circulation issues are probably just kind of a factor of the hypotonia and lack of movement. And it's one of the things that I plan on asking families about when I talk to them this year. In my opinion, that and this is partly going to a different study of a gene therapy trial that I'm a part of for a different disease. And when we increased movement, the circulation problems went away. And so that's my biggest guess, is that there's just not as much spontaneous movement. But it's something that I do want to look into. The dysautonomia, I think, is a completely separate issue, and I think is something that's unique to this disorder that I don't completely understand.
Can you define what dysautonomia is?
Oh yeah. Sorry. Dysautonomia just means that so autonomic function is the regulation of like blood pressure and heart rate and some of these kind of normal things that keep you alive. And when we talk about dysautonomia, it just means that there's a dysfunction or that there's a lack of regulation of blood pressure and pulse, and I think specifically in these kids is of temperature, where kids run a fever even though there's no infection. And I've seen this in kids even when they've been paralyzed in the ICU. So I think it's something that, and it's something that I don't see in other conditions. And so I think it's, I mean, we do see in other conditions, I shouldn't say that, but it's not as common. We don't.
One point about that, so G-alpha O is definitely expressed in the autonomic nervous system. Dr. Galligan in my department has actually done some studies in GNAO1 in the nerves in the gut, but also it's present in the SA node in the heart, and which is involved in autonomic regulation of heart rate. So it doesn't surprise me at all that there may be alterations that in G-alpha O function in the autonomic nervous system that may underlie some of these special properties that you're seeing.
Okay. Two last questions before we wrap up. Dr. Viehoever, there's just some comments I missed, sorry. Dr. Viehoever where did that one go? Sorry, what about the cardiac? Is there a risk of cardiac issues in GNAO1 patients? Is the heart like a muscle similarly impacted by low tone and things like that?
So cardiac muscle and other kind of like, when you think of like other, is a different type of muscle than say your muscles that control your limbs. And so when we think about low tone, we don't necessarily think about the cardiac system. And so, but what I don't know is whether or not there are risks for like sudden cardiac death that are associated with what Dr. Neubig was just talking about, like with, problems with the autonomics regulation, as in regulating heart rate and blood pressure and stuff like that. I think that there's not a huge amount of data to suggest that there is, but I can't completely rule that out. But no, when we think of low tone, it doesn't necessarily translate to the heart per se.
A follow-up on the dysautonomia that I think is interesting. Is there any treatment known for dysautonomia?
I mean, there are things that we try. To be honest, I haven't really found anything that has worked particularly well. But yes, there are things that and medications that we definitely do try. But
depending on the case.
Yeah.
One interesting question to follow up on that, that maybe your registry could answer. Although I explained how complicated the mouse models are with respect to gain of function, loss of function, if there were a particular clustering of dysautonomia in the movement disorder patients or in the epilepsy patients, that might give clues about what the mechanism is. So I think that's a question that'd be valuable to ask from within.
Okay.
Dr. Axeen, somebody asked, would frequent falling be considered a movement disorder or more likely drop seizures?
Good question. That keeps me in business and why I have a job is because it's very, very difficult to distinguish sometimes. For example, children can fall because they do have low tone or maybe not as much strength, or they have some of the abnormal movements, they get off balance. Drop seizures are very different. That is a electrical storm in the brain that actually causes that loss of tone. Sometimes I can distinguish that by just asking the families questions, sometimes by video, but oftentimes EEG is needed, and that's kind of the gold standard for distinguishing those two. So it really depends.
Okay, and last question. We got some more now. Dr. Viehoever, do movement disorders always get worse or can they stabilize?
Unclear. And then I think that is what I'm hoping to learn with this natural history study. I think if you had asked me this question, a few years ago, I would say it gets worse. But now it's clear that there are some kids that don't get worse and that they stabilize, and that the movement disorder is much more varied and complex than I think we first understood. And when and if the movement disorder gets worse and causes decline is also, I think, very varied. So I'm hoping that you guys will help me answer that question.
Okay, and then one more question. if G protein is so highly expressed in the brain, how is vision, hearing, intellect may be preserved? I don't know if Dr. Neubig or if we have any takers on that one.
Well, there are about 20 different G proteins. And so, you know, a change in one will still leave a whole lot of others that are functioning. And there's so many other receptors that don't even use G proteins. So it's just one element of a very complex picture. And so in particular, so knockouts of GNAO1, there's also GNAI1, 2, and 3, which are very closely related proteins. And so the redundancy of the brain allows a lot of things to happen, even when some piece of it is perturbed, I would say.
Okay, well, thank you all. Does anybody have any closing thoughts before we wrap up? And we could, I think, talk all day. Obviously, we're going to have an entire conference that we didn't get to be together, but we are looking forward to next year. But in respect of everyone's time and needing to take care of your families and everything, we'll go ahead and wrap up. Any closing thoughts?
Yeah, I just wanted to say thank you to everybody, and that we're going to do our best to, you know, continue this project and move forward in as much of the ways that we can. If for no other reason, to continue to gather information, but then also, you know, to apply for more funding and, you know, looking for bigger grants and stuff. So we are very grateful for all the families that are willing to help us. I can only, you know, imagine what it's like to take care of a child with special needs. And so, you know, everything that you guys are doing, we appreciate. And so I'm hoping that I'll have more answers for you next year.
Thank you. Well, to all of our attendees and families listening in, thanks so much for taking the time to be with us this evening. We're going to include a recording of this webinar and the McNamara Habib family webinar on our next monthly update. We've also had a couple people asking how they can participate in the registry and the natural history study, so we'll include links to that information as well. To our panelists, thank you all so much for caring about our GNAO1 families and for the hard work that you're doing. We sincerely appreciate it, and we hope everyone stays well during these times. Thanks, everybody.
Thanks very much.